because of the limitation of land resources. So it's a lot of things that probably many of you are familiar with, but being more selective in our crops that we plant in certain regions to be more appropriate for environmental conditions, using either selective traditional breeding methods or genetic modification to create heat and drought resistant or drought tolerant varieties of plants and animals, and being better stewards of the land when we manage fertilization, erosion, and weed control. So certainly there's a whole lot of work going on the crop side. On the livestock side, again, selecting livestock species that are more suitable for a particular area, its environmental condition, balancing how we use wild-caught fisheries and farm-raised fish production for both animal and environmental health, and then maintaining health with decreased use of medications. That's really more about antimicrobial resistance and transfer of antibiotic resistance from animal systems potentially into human systems. Where we focus at EnviroFlight and a number of other companies as well is kind of on alternative proteins. So there's a lot of novel proteins that are being considered and let's say novel nutrition sources that are being considered to augment our supply of nutrition for plants, animals, and humans as animals. And that may be plant-based selection. So I think in Canada, there's been a whole lot of work done on pulses and more, I would say, novel plant sources or less traditional in the recent years of agriculture. And then globally, there's been a lot of focus on alternative protein sources that are not necessarily plant or terrestrial plant derived. So the primary sources that are being looked at for alternative proteins are insects, and obviously we'll do a deep dive on insects. Algal proteins have received a whole lot of interest, particularly because algae can make long-chain omega-3 fatty acids, and that's one of the big drivers of creating a true sustainable alternative to fish meal and fish oil, is having something that has those long-chain omega-3s. Single-cell proteins are being looked at, both bacterial and fungal protein synthesis. And then obviously, as I mentioned, plant-derived proteins from novel plants or isolated proteins from kind of more traditional plant species. And then cell culture-based proteins, which are really much more focused on the human nutrition side, but we're starting to see some, let's say, R&D going into creating cell culture proteins for animal consumption. There's been over $2 billion invested globally in insect agriculture in the last 10 years. And I just give you a snippet of a few major facilities. So this is Aspire. It's a cricket production facility in Ontario or in London, Canada. This is a Nova Feed in France. This is a black soldier fly larva facility. And this is EnviroFlight. We're based in Maysville, Kentucky. We raise black soldier fly larvae. I like to throw the pictures up there to give a sense of scale, because these are massive facilities that are coming online. These are not, kind of very small kind of garage, backyard types of operations that are being funded. And part of that is because we know that scale is required to achieve commercial viability for these industries. There's been a whole lot of government support for these industries as well, primarily in the EU and Asia and in Canada, where government agencies are supporting the development of these novel technologies. Why are we focused, and my company and numbers are focused on insects, is for a variety of reasons. So first is that virtually every animal that we raise for meat, milk, or eggs have some insectivorous part of their natural feeding strategy, whether that's part of their life stage. So say salmonid species, trout and salmon that are naturally insectivorous when they are in their kind of young starter life stages, to what you might not consider an insectivore, but a traditional herbivorous species like a cow or a horse, where every bite of grass that they take contains insects. So there's some natural evolutionary physiology that it goes along with feeding insects to animals. Additionally, there's some really exciting opportunities for creating really high amounts of protein or nutrition on a per hectare or per acre basis, partly because of the life cycle of insects. We'll talk briefly about how rapidly they propagate and grow, but also because we can farm in 3 dimensions. So this picture here is a picture of our grow out operation, and really the limit to how high we can grow is based on engineering and kind of operational efficiency and safety. But you could go, if you saw the picture of Aspire, I think they're 100 feet or something tall facility for growing crickets. So you really have the ability to farm in a 3-dimensional space, which dramatically improves the yields per area. And then the final piece is that insects are generally 100% edible. So unlike, say, soybeans that we have to dehull, or poultry that we need to remove feathers, or beef or pork where we need to remove hides and hooves and things like that, the entire insect is edible. So you put those three things together: really rapid life cycle, which means we can have a lot of generations in a single year. Vertical farming potential, and very high edible yield relative to other protein sources, you get this massively increased potential yield on a per acre, per hectare basis. And I'll just note that these data represent published literature. In commercial operations, we're seeing 500,000 pounds of protein per acre per year come out of insect production facilities. So there's this great opportunity to create really high amounts of large volumes of material in a smaller land footprint. It's also sustainable because insects are cold-blooded, so you don't have they don't have to put as much energy into thermoregulation as traditional livestock species, and so they can be more efficient at their use of energy. And because we can farm vertically, and because of their really rapid life cycle, our life cycle assessment, the assessment of the impact on the environment, on human health, on things like your natural resource usage, generally reflects an improved sustainability coefficient, depending on which metric you look at, when you raise insects compared to traditional ingredient sources. So the data here represents our internal LCA that we did with the University of British Columbia. There are lots, for those of you who love LCAs, there's a lot of different ways to do them. The data I'm presenting here represent mass-based, where you allocate the cost based on every piece in and out by a mass base, how much you use, how much you make, or an economic-based LCA on my right, which is more about the value of the inputs and the output materials. Either way that you run the models, you can see that this is our BSFL based meal. We have dramatically lower carbon footprint than traditional livestock ingredients, or traditional protein ingredients, including plant-based protein ingredients. That's not to say that we're better. I think what it means is that insect ingredients provide an opportunity to have a reduced carbon footprint and help offset maybe the cost of producing things like chicken and beef that are going to be produced because the human diet wants, people want to eat those protein sources. But if we can feed those animals with something that reduces their carbon footprint, then we can overall be more sustainable in producing protein and energy. So the three primary insects that are being commercialized, I mentioned crickets and black soldier fly, and then mealworms are the third. So black soldier fly by far is the biggest in terms of number and dollars that are being invested in commercialization. Mealworms, there's a very large producer of mealworms in France called Insect, and they have some facilities, a small facility in the United States, and also a couple others in other countries in the EU. Mealworms also are farmed extensively in Asia. And then crickets. Crickets have received maybe a little less focus and attention. They're a little more difficult, I would say, challenging to grow and a little bit more restrictive in their dietary habits, but they are, I'm going to call them the gateway insect for human consumption. So when we think about getting the Western world to think about consuming insects directly in your diet, in a taco or in a cookie, crickets tend to be a more palatable ingredient for humans than the larval stage insects that remind people of squishy, icky things. So crickets really, the cricket industry is really focused a lot more on human food applications and maybe pet food as well. I showed a figure here that shows the life cycle of black soldier fly larvae, and they are probably the fastest growing of the three species. So an adult fly will lay somewhere between 500 and 900 eggs in one clutch. Those eggs hatch in 2 to 4 days, depending on the environmental conditions. And those eggs, those larvae, when they hatch, grow exponentially for the first 2 to 3 weeks of life. Most companies that are harvesting black soldier fly larvae to create ingredients are harvesting right around this life stage. They go through multiple instars. Essentially, they molt and then get bigger. They shed their exoskeleton and then grow again. And so this is generally the 4th or 5th instar is going to be chosen for harvest for an ingredient production, because that's where you have the highest digestibility of nutrition from this insect. As the insect goes through the later instar stages, it actually creates more chitin and sclerotized protein that forms that hard exoskeleton that protects the insect when it goes through complete metamorphosis to become a fly, but it also dramatically reduces digestibility. And so most people are not harvesting for ingredient production at these later life stages because it just has very low digestibility. And so in our production system, about 95% of the larvae go to that 4th or 5th instar and become an ingredient, and about 5% become broodstock and continue the life cycle. What's neat about the adult black soldier fly is that they have like a sponge-like mouth, but really it's designed for drinking. In the wild, they will consume nectar, but otherwise it's water and nectar. They have no functional mouth parts, for example, for chewing. So there's really essentially very little nutrient requirements of an adult fly. They hatch or they emerge with a fat vessel that supports their energy requirements for that very brief life stage, a lifespan where they breed, lay eggs, and then die. Oops, sorry, too fast. So when we feed those insects, we have the really cool thing about black soldier fly larvae is they are omnivores. We can feed them with lots of different feed ingredients, and that allows us to upcycle ingredients. And when I say upcycle, I mean we take things that have low value to either human or animal nutrition or no value, they're being landfilled or disposed of directly, and we turn them into higher value in protein, energy, and nutrition sources. There are restrictions in what we can feed black soldier fly larvae based on AFCO, the American Association of Feed Control Officials, and your agency, CFIA, has similar requirements generally for feeding restrictions. So I've got a couple of pictures here just to illustrate some of the things that people have focused on for feeding insects. So this would be pre-consumer food waste. So this is the waste that comes either from growing and processing of fruits, vegetables, bread, bakery items, whatever the case may be, but it never makes it to a consumer in a retail setting or grocery store or restaurant. This is readily used in black soldier fly production. And in fact, there's a facility in Quebec that's coming online right now that really is focused on pre-consumer food waste and diverting that from landfill. The difficult thing about pre-consumer food waste is it's really variable. So those of you who are nutritionists understand that you can imagine maybe how hard it would be to get consistent nutrition when you have this huge range of ingredients. So it's kind of an art in and of itself how to balance the feeding program when you use pre-consumer food waste. Post-consumer food waste would be something that's already made it to a table or is in the salad bar at the grocery store. It's been potentially handled and touched by humans. That generally is not allowed right now as a feed ingredient for black soldier fly larvae because there's some safety concerns. So if we have introduced something into that food chain, bacteria, viruses, something like that, mycotoxins, then there's concern about what may happen in the insect. So there's some research being done right now to kind of validate safety of post-consumer food waste because that does contribute to a whole lot of landfill waste. This picture is distiller's grains. This will be a wet distiller's grains, but there's a lot of brewery and distillery and ethanol production waste that is being fed to black soldier fly larvae, either in the dried form or in the wet form or truly like the liquid stillage form. They really, really like sugar and they really like alcohol residues. So they're very happy larvae when you feed them with distillery and brewery waste. There's folks looking at offal from the aquaculture industry. This is a great way for black soldier fly larvae in particular to bioaccumulate omega-3 fatty acids. It's by feeding them things like salmon offal. So again, things that would go into landfill or waste streams, but provide really great nutrition for black soldier fly larvae. And then the final picture is manure. So there's a whole lot of literature supporting the ability of black soldier fly larvae to use manure from most species pretty effectively. Right now, there's very little regulatory approval for that, and that's because of the safety concerns of, again, more pathogen food safety kinds of concerns, that if you want to use manure, you're really going to have to document that there's no risk of carryover of a pathogen or some sort of residue from a diet, like an antibiotic residue, for example, that would transfer into the black soldier fly larva. But I think hopefully I can illustrate here the vast diversity of ingredients that we can use to feed these insects. It's important to know that feedstock can influence the composition of the insect, particularly the fat to protein ratio or kind of caloric profile. The fatty acid profile is in many cases kind of you are what you eat with black soldier fly larvae and mealworm larvae. Vitamin content, particularly the fat-soluble vitamins, are directly related to what's in the diet. And then black soldier fly larvae in particular, the mineral content is related to the diet. So they use their exoskeleton kind of to remediate excess minerals. So if you feed them a diet high in, say, cadmium or lead, you're going to get a black soldier fly larva that's high in cadmium or lead. So we really have to be mindful of the feedstocks and what potential nutrient content or let's say anti-nutrient content might be there and how that might impact the composition of the ultimate ingredients. The data here just represents or shows the fact that these insects are both mealworm and black soldier fly larvae are really good at remediating mycotoxins and mold. They grow naturally in their wild type, particularly black soldier fly larvae in their native environment would be in like a compost pile where they'd be exposed to mold. This trial demonstrated that if you spiked their diet with aflatoxin B1, black soldier fly larvae essentially remediated 80+% of that aflatoxin B1, and it was only at the really high levels that anything was detectable that was not completely remediated. Yellow mealworms were even more effective at remediating mycotoxins, aflatoxin B1 in particular. This is really exciting because there are obviously federal limits in both Canada and the US for how much mycotoxin contaminated grain can be fed to livestock. And so that's also a huge waste stream seasonally when you have corn or wheat or something like that that has high mycotoxin levels. So there's a lot of conversations going on right now about how black soldier fly and mealworms could be used to essentially remediate those grains to allow us to produce high quality ingredients and not just landfill things that have unfortunately become moldy. Most of the rest of the conversation and most of the rest of my slides are really more about how we can feed these ingredients to animals. But I do want to mention that, you know, this industry is rapidly growing, and we are absolutely thinking about things like husbandry and animal welfare as we think about how we raise these insects at scale. So I kind of joke we're like 1920s chicken farmers, right? We're figuring out how to optimize our systems. We're figuring out what the best practices are for raising these animals. We're driving research in academia to help understand, you know, the big questions that we don't have answers for. We have a trade organization that represents North America. It's really US and Canada focused at the moment. And we've just published the best practices guideline to help producers of insects understand at least what we currently know about opportunities and important things you need to consider when you're raising insects, like how to maintain your feed in a safe situation so you're not exacerbating food safety risks, how to process insects, what are the opportunities to create ingredients. And we're currently tackling some big heavy topics now as well, like insect welfare. And then frass is our biomass, and I'll show you a picture of that, and kind of thinking about that as a natural fertilizer source. So if any of you have any interest in insect agriculture, I encourage you to go to the NASEA website and learn more about that. Lots of opportunities to get engaged with us as we figure out kind of where the industry can go, where our gaps are, and what we need to learn. I spent a few slides on nutrient composition, and again, all this is readily available, and if you want more information from me, please feel free to reach out. But in a nutshell, those larval stage insects, the ones that look like little caterpillars, they're fatty. So they tend to have lower protein and higher fat than insects that have a hard body exoskeleton like crickets. So you'll see that reflected here in the crude protein content of either the whole cricket or a partially defatted. Crickets tend to be much higher protein and lower fat. Mealworms are kind of in between, where you get your higher fat because they're a fattier larval stage insect. And black soldier fly larvae have the highest concentration of fat of crude lipid of any of the insects that are being commercialized. As I mentioned, black soldier fly larvae in particular will accumulate minerals in their exoskeleton, so they tend to have higher ash content. But they also tend to have a little lower chitin. So as a nutritionist, when you're thinking about whether your animal that you're feeding has chitinase and is adapted to eat chitin, there's some difference in the chitin content of each of these insect species. All in all, really great energy sources, either because they're really rich in protein or more rich in fat, but generally very available, digestible amino acid, fatty acid profiles. Again, I'm not going to go through all these data points, but what I want to show is that of the 3 insect species, the amino acid profiles and kind of the ratio to crude protein tend to be similar, which is not surprising. Their tissue composition is relatively similar on a protein basis. And the digestibility of amino acids tends to be really good. So there are impacts of processing on ingredient digestibility, but in general, we see availability of amino acids that's as good as or better than things like soybean meal or high quality poultry meal. And maybe not quite as digestible as high quality fish meal for at least a few of the amino acids. And by the way, all these data are in cecectomized roosters. So these are I would say pure amino acid digestibility numbers, not total tract digestibility. I won't, again, spend too much time on this, but just to say that insects do contain a whole lot of other essential nutrients. We generally think about them as protein and energy sources, but they can be, all of the check marks represent meeting NRC requirements for those respective poultry, dog, or cat requirements. And you can see for most nutrients, most of the minerals, insects in general will meet the nutrient requirements. Calcium is the one that's unique for BSFL. They do have generally a positive calcium to phosphorus ratio. Mealworms and crickets will be an inverted calcium to phosphorus ratio. So if you're using mealworms or crickets as a primary food item for, say, reptiles, you have to be very aware of that because it's an inverted calcium phosphorus ratio. You'll have to provide additional calcium to get that to be a appropriate 1-to-1 or higher. Oop. The vitamins, again, as I mentioned, the fat-soluble vitamins tend to be enriched if the diet is enriched. There is some evidence that mealworms can synthesize vitamin D3, which is in and of itself super cool because they're normally like not exposed to UVB, but if you expose them to UVB, they make D3. So pretty cool insect from a kind of a basic biology perspective. You can enrich all of them in carotenoids by feeding dietary carotenoids. And then the B vitamins are generally, it meet all the NRC requirements in these species. What you'll notice that's not on this list is vitamin A. Insects don't use retinol the way that higher order animals do. So you just don't have a source of retinol or kind of retinoic acid in insects that will meet nutrient requirements of any species. You can gut load these if you're feeding live insects. You can provide some vitamin A in the diet and essentially fill their GI tract to get some vitamin A into that bug. But at this point, there's really no way that we know of to enrich the actual insect with true retinol or retinol palmitate or retinol acetate. And then fatty acids, generally linoleic will be in adequate concentrations in any insect species. Linolenic is really generally diet derived. So if you feed it or diet rich in linolenic, you'll see it in the insect, and the same with the long chain omega-3s. So if you feed long chain PUFAs, you can get them enriched in any of these species. BSFL in particular make lauric acid, which is a C12 saturated fatty acid. It's found naturally in coconut and palm kernel meal or palm kernel oil. Really interesting fatty acid. I think I have another slide, but has some antimicrobial and antiviral properties. That is based on the carbohydrate content in the BSFL diet. So they actually synthesize that fatty acid in response to their dietary source of carbohydrates. So I'm going to go through the next slides really quickly, but again, if there's questions at the end, I'm happy to answer them or provide references. But when we think about insects, most people are thinking about traditional alternatives to traditional protein, amino acid, and energy sources. But there's also some value-added opportunities that I'll focus on at the end because of lauric acid, because of chitin, and because these insects all make antimicrobial peptides, which essentially their natural immune defense, and turns out are active in the finished ingredients. So lauric acid, really quickly, again, C12 fatty acid, saturated fatty acid, and has quite a nice literature basis for having antiviral and antimicrobial activity. Most of the early work with lauric acid was done using purified lauric acid, and it's really unpalatable, so you have to force feed animals to get them to consume it. BSFL or coconut or palm kernel oil give an opportunity to provide it in a more natural form that tends to be very palatable. And all of these data in this table just reflect changes in bacterial abundance, either in the microbiome, or the gut microbiome, or systemic disease challenge situations in a variety of animal species in response to the complete ingredient, but highly enriched in lauric acid. Chitin, again, is an interesting component of insects. It's found in their exoskeleton, and it can be a source of of nitrogen and energy if an animal has that has chitinase enzymes. So poultry, for example, have the enzyme and it can be upregulated. So maybe in the first week of feeding insects, they may not be generating nutrition from chitin, but you'll see that enzyme becomes upregulated quite quickly. Other species may not have chitinase, or it's really low activity. And if you don't have chitinase, then that chitin essentially acts as a prebiotic or fiber source. And we also know that it can kind of prime the gut immune system for an anti-parasite response. So there's been differential responses to feeding chitin in terms of nutrition and kind of digestibility of ingredients. And most of those tend to be like a dose response. So you kind of get a little bit of chitin, you get some beneficial effects, you start pushing it really high, and you start seeing reduced digestibility and reduced feed conversion ratios. So it's what we don't know maybe is the perfect amount of chitin for each species, and that's something that I think folks are working on right now. And then, as I mentioned, antimicrobial peptides, these are the innate immune defense of insects. So if you think about them, they don't have very complicated, antibody responses and anti-cancer responses and those kinds of things that higher order vertebrates will have evolved to have. What they do have is a whole bunch of these tiny little peptides that are antifungal, antiviral, antimicrobial, and they not only excrete them into their environment, but they're also found systemically. So if you take a blood sample from these insects, you will find these peptides. What's really cool is that they're tiny. So the risk of antimicrobial resistance is really low because they're just such small molecules. They're not really big, like a penicillin molecule that's much easier for a bacteria or virus to generate resistance to. And they also really cool, survive processing. So as we take that insect and we create a dried ingredient from it, we see antimicrobial peptides can maintain activity. And so we're seeing health benefits in animals fed insect-derived ingredients that are likely related to all three of these: antimicrobial peptides, lauric acid, and chitin. And I'll show you some data towards the end of the presentation. Going to go briefly through some of the data that were generated to get regulatory approval. When I started with EnviroFlight in 2017, we had no regulatory approval, so insects could not be fed to any livestock species, not to pets in the United States. In Canada, pet food manufacturing is regulated a little differently, but essentially the slate was clean and we had to prove safety. So generally you prove safety by doing a dose response feeding trial. You show that there's no negative impact of the ingredient on the animal through blood work, maybe tissues if it's a production animal, maintenance of body weight, feed intake, those kinds of things. So a whole lot of work has been done to demonstrate safety of these ingredients in a lot of different species. First, there was work in live larvae feeding to poultry, and really showing that there's no significant difference in performance in terms of your traditional metrics like feed conversion and egg laying. But significant improvements in feather condition, because those birds, instead of plucking each other, were foraging for insects. Subsequently, there's been a lot of work showing that you can also improve skeletal integrity of animals being fed insects, and it's probably more of a changing their behavior and inducing motion and movement because they're just moving more, and so you get better calcification of the skeleton, and that's been shown in pigs and poultry. When we move into the dry ingredients, what we've done, EnviroFlight's done a whole lot of the regulatory work. What we did was fed graded levels of either our protein, our fat, or our whole larva, and this, I'm only showing you the high end of each of the inclusion rates, and did those graded levels, and then demonstrated that animals performed well or relatively consistently to controls, and then again the blood work and kind of CBC clinical chemistry showed that there was no health impact. What I want to point out here is, first of all, when you get these really high, I would say higher inclusion rates of the protein-rich meal, we do see slight reductions in feed efficiency and yields performance, whether that's weight gain or, in this case, egg yield. And again, we think that's probably a chitin-related impact, where you kind of gotten to the high level of the ability of the animal to use chitin as a nutrient source, and most likely is starting to be a fiber and impairing or reducing digestibility of other ingredients. You don't see that impact on the oil. Oil performs as well as soybean oil and in other projects, poultry fat. And the whole dried larva generally performs really well because you're getting that energy in the protein source. The other thing I'll mention is that our diet is based on distillers dried grains, which is a great source of carotenoid pigments, and we see that those carotenoids are available to the laying hens that are eating those ingredients and translate to enrichment in egg yolk color when poultry or laying hens are fed meal or oil. Oops. We also did safety work in broiler chickens. And so I'm not going to present the data here, but I wanted to show these data because I think they're really interesting. In this trial, the broilers were fed 1%, 2%, or 3% whole dried larvae mixed into their pelleted diet. And then we're challenged with a fowl typhoid live clinical disease challenge. And this is what you see here is a significant improvement in survival or reduction in mortality associated with these very low inclusion rates of these ingredients. This has been consistently shown in a variety of species, and again, likely related to the antimicrobial peptides and lauric acid content. It's clearly not a nutritional impact at 1%, 2%, or 3%. This is something that's mitigating animal health concerns. And I'll show you a slide in just a few slides here that kind of hints at what we think might be happening in these animals. We've done work in pigs. From a regulatory perspective, your model would be in nursery pigs, so up to about 42 or 43 days post-weaning. And again, demonstrating that we can successfully feed, this is oil, to baby pigs with no impact on health. And in fact, in baby pigs, the lauric acid appears to be more available as an energy source than both poultry fat or soybean oil, and we see that in improvements in body weight gain without increases in feed intake. So feed efficiency generally is improved in nursery pigs. We've done this in, I would say, research scale, super clean environments, and then also semi-commercial, so a little bit more stress, traditional commercial environments, and we've replicated this a number of times. So for whatever reason, young pigs seem to be really able to use lauric acid as an energy source in the gut, and that translates to improved yields in the nursery phase. We don't see that past the nursery phase. It seems to be really about the developing gut. I didn't spend a lot of time talking about the biomass that we create in our feeding program, but when we raise our insects, we get the insect, but there's biomass, just like any other animal production or ingredient production operation. We have a byproduct. Our byproduct is called frass. It's really the remaining feed, hopefully not much if we're doing a good job with managing our feed ratios, but any undigested feed, the digested feed and excreta, and the exoskeletons from those subsequent moltings that the larvae go through as they go through their growth stages. That product is called frass. In the United States, in Canada, it can only be marketed as fertilizer. In the United States, it can be sold as fertilizer, but it also can be sold as a feed ingredient. That's because we heat treat it. It's got no food safety risks, and it's actually, as we've learned, a really interesting feed ingredient for animals, particularly those that are more omnivorous. That this represents more of an omnivorous type of, not a true animal protein. And so these data represent a trial that we did. We call that product EnviroFeed. We did a trial in nursery pigs using 3 or 6% inclusion of EnviroFeed. Diets were isocaloric, isonitrogenous. And what you can see here is our average daily gain up to 14 days was essentially the same, with maybe even a slight improvement in the 6% frass inclusion. So again, these were formulated to be isocaloric and isonitrogenous. Our hypothesis for the slight improvement in yield, and we've now replicated this trial with a bigger cohort and seen a more statistically significant response. Our hypothesis is that a lot of those things that are in the insect are also in the frass. So the antimicrobial peptides, again, the insects secrete them into their environment. They kind of farm their environment, if you will. And those exoskeletons that contain chitin, a lot of those would be in this frass. So it's likely we're providing chitin and antimicrobial peptides along with good nutrition to these young animals and helping them grow during early developmental stages. We've also done a whole lot of work in dogs and cats, including some with your team at Guelph. But we initially in pets, obviously, we don't do a whole lot of invasive research, but we do just standard digestibility, palatability, acceptance, fecal scores to ensure that there's not significant changes that a consumer would see if these ingredients were included in dog food or cat food. And then you will do clinical chemistry, CBC, so slightly invasive blood work in dogs and cats. What you can see here is this is our control diet, which is a poultry-based diet, poultry meal and poultry fat. And this diet is 20% Enviro meal, our BSFL meal. Again, we fed graded levels. I'm only showing the data for the highest inclusion rate. And then this is Enviro oil. And for those of you who are companion animal folks, this is externally applied on an extruded kibble, and the meal is internally applied, obviously, in the extruded kibble. And what we see is similar dry matter digestibility, similar fat digestibility, and a slight reduction in protein digestibility, again, as we're kind of hitting those higher levels of inclusion of the protein-rich meal that has a reasonable amount of chitin. So we're likely seeing, we believe, a chitin impact here on digestibility of protein. See the same response in cats. Generally no change in fecal scores in dogs or cats. You'll notice here we only went to 2.5% oil in the cat model. They really did not like 5% oil on an extruded kibble. We've subsequently, and I don't remember if I have a slide or not, but done work with retorted diets, and they really like the oil in retorted diet. So it may be more of a texture issue than a palatability of the oil per se. But in this trial, we only went to 2.5% for oil. Again, you'll see this slight reduction in protein digestibility associated with that higher inclusion rate of the meal. And cats have no chitinase activity, so that's not surprising that we see maybe a more dramatic response in cats than we saw in dogs. This is the retorted diet work that we did. We did this in cats. So this says these, keep in mind, these look like lower inclusion levels of these ingredients, but it's on an as-is basis. So this is actually about 30% protein on a dry matter basis and about 6% fat on a dry matter basis. We also did the whole dried larva. We ground it up because nobody wants to see little bugs in your cat food. So it's all ground up in a homogeneous form. But you can see our meal, again, we have this slight reduction in protein digestibility. This is not as dramatic when you do the whole dried larva because it doesn't have as much of the chitin and the protein. And then our oil digestibility is really good. You even see an improvement in fat digestibility. I mentioned the preference was actually greater for the BSFL enriched diets than the control diet. So in all three, the cats preferred, and the retorted diet preferred the insect derived ingredients to poultry-based ingredients. All right, I want to be mindful of time, so I'm going to zoom through the last couple real quickly. We have been, a lot of our work has been focused on the regulatory side of it as we worked on regulatory approvals, but as we've kind of moved through that process and we are close to being done with regulatory approvals, we've started to look more on these health benefits. And so the data here represents some work we did at University of Idaho looking at trout. So we fed trout 2 control diets, either a soybean meal control or a fish meal control. And then we added either 2.5% or 5% whole dried larva or 2.5 or 5% meal. That the diets, again, were formulated to be isocaloric and isonitrogenous. So this is not on top of those other ingredients. And all of the BSFL ingredients were added to a soybean meal enriched diet. So the fish meal, we did not add to a fish meal based diet. With the idea that soybean meal obviously is much less expensive than fish meal, but soybean meal causes pretty dramatic enteritis in trout, which you can see here. If you like histology, these are really exciting slides. That's a really ugly gut, very inflamed, and that's just a trout-fed soybean meal. Thank you for smiling at my comment about liking histology. What we see is really cool is that when we fed our whole dried larva to a soybean meal-based diet, and this is just a control, a healthy or an animal just during the feeding challenge. You can see essentially we mitigated all of that enteritis with a very low inclusion rate of the whole dried larva. And then we did a disease challenge. So we challenged them with a flavobacterium challenge, which causes early hatchery mortality quite significantly in commercial operations. And what you see in the histology here, this is post-challenge in the soybean meal fed fish. You can see their guts look even worse. A lot of inflammatory cells, not a lot of surface area to absorb any nutrition. And again, in the fish that were fed that whole dried larva, the guts still look quite healthy. There's clearly some inflammation. There's clearly they're not as pretty as those let's say non-challenged animals, but the guts clearly are still able to digest and absorb nutrients, and that's reflected in mortality. So in our soybean meal fed control animals, we had almost 65% mortality to that disease challenge, and that's about 30% mortality, 35% when we get either 5% of meal or 5% of whole larva added to the diet. We also see significant changes in the microbiome when we feed those animals those different diets. So in orange, these are fish meal fed trout. The green represents the soybean meal fed trout, and this cluster up here in the blue that I've circled in blue represents all of the insect fed animals. So again, pretty interesting. These are relatively low inclusion rates of ingredients to cause pretty dramatic shifts in microbiome. And we've now seen this data that not only similar shifts, but similar taxa of bacteria that are shifting in cats as well. A paper was just published, and we've got some internal data supporting the same kind of bifidobacteria populations being increased, lactobacillus populations being increased in insect-fed animals. Just give you an idea of where we are on regulatory approvals. So this is, again, AAFCO, the American Association of Feed Control Officials, but CFIA generally follows AAFCO guidelines, with the exception of the fact that pet food is not regulated the same way by CFIA. But you can see we've got, in the United States, we've got almost everybody approved. We've got cats in progress. Those dossiers are under review with the FDA. And we've got some expansion on our aquaculture definitions. So I would say by 2025, we'll be done with definitions for everything except ruminants. So there's no ruminant or equine definition yet approved. And to my knowledge, no dossiers in for those species at the moment. I will finish up with just telling you a little bit more about EnviroFlight. So we started in 2009 in Ohio. The founder was very interested in biofuels and more sustainable alternatives to wild fisheries. We had a pilot plant there that Darling Ingredients invested in, allowed us to scale up to a true pilot scale. In 2018, we opened our production operation in Maysville, Kentucky. We're the first in the U.S. To do it. There was a company out of Calgary that was in the business at the same time. So I think their plant might have opened a couple days before ours, but 2018, 2019, we were seeing commercial scale production. And then in 2020, we doubled our facility. In 2022, we closed that facility in Ohio and moved down to Raleigh, North Carolina, where I am now. I mean, this is where our innovation center is, where all of our R&D, sales, marketing, kind of corporate functions happen. And really, I know it's 2024, I need to change that, but we're really looking now at kind of where we expand based on our regulatory market opportunities. We do focus a lot on innovation here. So if you have interest in insect research, we've got a great team here doing lots of cool things with genetics, nutrition, animal biology, and animal husbandry, and immunology focused on those health benefits that we talked about. Also regulatory, it's a never-ending process. So as we finish AAFCO approvals, then we'll worry about how to export ingredients to other countries and validating safety with those markets. Ooh. So I wanted to make sure I left a couple minutes for questions. So I appreciate your attention. Hopefully insects are super exciting to all of you now. How can I answer any questions?
Amazing. Thank you, Liz. That was fantastic. I think Kate is here, but I don't know about her connection, so I'll just keep forging ahead. Does anyone have any questions? A lot of questions for Liz. Excellent. So if you'd like, you can also turn on your camera and unmute your microphone if you want to ask questions. I can call on people, or feel free to type them in the chat and I will read them out to Liz. Yeah, Dave, go ahead.
Hi, Dr. Cosmos. Kotsos, I hope I'm pronouncing that correctly.
You're
Can you
good.
hear me okay? Yeah, good. So my lab does a lot of work with feeding insects, especially black soldier fly, to rainbow trout. So it's nice to see those results. And we are finding similar effects on like lactobacillus and things like that going up in the gut microbiome, and really cool things looking at the oil and the chitin side of things. I actually have a few grad students working on this area, and one's going to defend in a couple months. So I'll definitely keep you in mind when that research is complete. I want to ask about Entera. Used to be a huge company, and a lot of labs at our department collaborated with them a lot. They're Canadian from BC, and so they're not a company anymore. Do you find that now there's opportunity to work more with researchers at Guelph, or do you think there's a space for more collaborations between